This is lecture 20 on digestive system part 2. We'll actually pick up where we left off from digestive system 1 with the esophagus. The esophagus is a simple structure. Its function is simply as a tube. It conveys food and liquids from the pharynx to the stomach. It is a hollow muscular tube, roughly 25 centimeters long, or about 10 inches long. It will obviously increase in length if the person has a longer torso or is taller. And its narrowest point is at the beginning. This is just posterior to the cricoid cartilage. Right here is going to be some of our major structure, the upper esophageal sphincter. Remember that a sphincter in itself is simply a band of smooth muscle that functions to control the flow of things through it, in this case food. It prevents air from entering the esophagus and responds to the process of deglutation when you swallow, where food is pressed up against the sphincter, thereby causing it to open and allow it to shift down. From there, it'll progress downwards to the lower esophageal sphincter, otherwise known as the cardiac sphincter. This is going to be at the inferior end of the esophagus, and it is normally contracted. Again, it's going to be a band of smooth muscle, and this is designed to prevent the backflow of the contents of the stomach into the esophagus. Here we can see a cross-section of the esophagus and its different layers. We can clearly see the lumen in the center, where the hole is, with a layer of stratified squamous epithelial. This is going to, of course, be non-keratinized. Remember, keratin is going to be on our actual skin. The stratified squamous is designed to help prevent excessive abrasion, or rather protect against it. From there, we move into the lamina propria, into the submucosal layer, and then into the muscularis externa layer, which is going to have those two bands. It's going to have both the circular and the longitudinal layers before we get to the outer serosa layer. Here we can actually see some different pathologies that occur from an esophagus. So in the lower esophageal sphincter, when it's healthy, is normally constricted, and it's going to hold stomach acid in. However, in some cases, we have what occurs is called GERD, or gastroesophageal reflux disease. Now, when you eat, it is normal for some acid contents to reflux into the lower esophagus. However, if the reflux happens too often, or if stomach acidity is particularly strong, then the damage to the lower esophagus can occur, and this results in the gastroesophageal reflux disease. Symptoms of this disease include heartburn, stomach pain, difficulty swallowing, persistent sore throat, as well as others, maybe indigestion consistently. This can be treated by behavioral or different medications and prescription drugs that block acid production, but over time there is a risk that normal esophageal cells will be replaced by intestinal epithelium. This is known as Barrett's disease. Again, this is from excessive acid building up over time, and we actually have, in this case, where some of the esophageal cells will turn into intestinal cells. Now, this is bad because it can actually increase the likelihood of developing esophageal cancer in many of these cases. We can also, of course, have hernias that occur, excessive inflammation. So these are a number of conditions that can occur in the esophagus. Next, we will discuss the stomach. Now, the stomach has a few functions. It is designed to store ingested food, mechanically break down food, chemically digest food with the aid of acids and enzymes, but do remember here that no absorption of nutrients occurs here. Some lipid-soluble substances can be absorbed, like aspirin and different drugs, and water can, of course, be absorbed through here as well. Now, the stomach has a highly variable shape depending upon its contents. When it's empty, it's a muscular tube with a constricted lumen, and when it's full, it can expand up to 1 to 1.5 liters of material. And this material is a combination of different acids, enzymes, as well as the food substance that was ingested, that it makes chyme. Chyme is going to be a viscous, highly acidic, soupy mixture formed from food, saliva, and gastric gland secretions. The stomach can expand larger, but it is at risk of tearing if it goes above the 1 to 1.5 liters. Now, there are two major curvatures, the lesser curvature that forms the medial surface and the gradial curvature that forms the lateral and inferior surface. Here we can see a diagram of the stomach. We've had to lift the liver up out of the way to be able to see it. You can see a few of the major structures. The esophagus is going to actually come down and insert into the lesser curvature. Projecting up from the lesser curvature, we have the fundus. The fundus is going to be a large bulge shape, and fundus, think fungus. The idea is that it's like the top of a mushroom. The greater curvature is going to be on the outside where we actually have the greater omentum. And you can see the greater omentum there, which is actually going to be a lot of fat and blood vessels that extend over the intestine just below the greater curvature. The greater omentum has copious adipose tissue deposits which serve three major functions. It cushions the organs, serves as an energy reserve for the body, and insulates the anterior abdomen from changes in temperature. Other structures to note on the stomach are going to be the main body of the stomach, and then the region that connects to the duodenum or the small intestine just below the liver and gallbladder, and that is going to be the pylorus or pyloric region. Here we can see a cross-section of the stomach and looking at its different layers. Now remember, in the muscularis externa, we typically have just two layers, the longitudinal and the circular layer. The stomach actually has an additional layer called the oblique muscle layer that hangs diagonally and will be the deepest of all these muscular layers. This is due to the shape of the stomach and allows for the action of churning, where we can actually start to twist and distort the stomach in order to compress and mix food more effectively. On the inside, we notice an increased surface area and a lot of ridges inside the stomach. This is called rugae. Inside the rugae are going to be different gastric pits that secrete a lot of the acids and enzymes that we'll talk about that aid in chemical digestion. The pylorus consists of the pyloric antrum, which is the entrance to this area, the pyloric canal, which would thereby enter into the pyloric sphincter. There are two sphincters on the stomach that would separate it from the other structures. From the esophagus, we have the lower esophageal or cardiac sphincter, and from the small intestine to the stomach is separated by the pyloric sphincter. Now, just to reiterate, the functions of the stomach are storage of ingested food, chemical and mechanical breakdown of ingested food, protection from different pathogens, and there's also the production of intrinsic factor, which we're going to talk about. This is going to be needed to absorb vitamin B12. I want to be very clear here that no nutrient absorption occurs in the stomach, as people often think. Nutrient absorption occurs primarily in the small intestine. Now, looking at some of the internal structures, we're going to zoom in on the rugae. As we go into the rugae, we'll look into the mucosal layer. In the mucosal layer, we actually have a simple columnar epithelium with numerous mucus cells. These mucus cells are essential in protecting the epithelial layer from the different stomach acids and enzymes. Now you can see as we extend deeper into the mucosa, we get into the gastric pits. Throughout this epithelium of the mucosa, there are numerous indentations called gastric pits. In here, we have stem cells at the base of each pit that regenerate epithelial cells that constantly replace those from damaged cells by stomach secretions. In the body and fundus, ducts of the gastric glands are found in the lamina propria, so a little bit deeper down. And these empty gastric secretions into the gastric pits and then into the lumen of the stomach. So here we can, as we've talked about, we have the mucus surface cells, which produce copious amounts of mucus to protect the lining of the stomach. We then have the gastric pits that will replace these cells, and then we have the mucus neck cells in between that produce mucus to lubricate food entering the stomach. As we go deeper down, we can see some of the secretory cells that are involved in the chemical digestive process. So these are some of those structures. The parietal cells. Parietal cells secrete hydrochloric acid and intrinsic factor. Hydrochloric acid is very acidic and kills microorganisms as well as aids in chemical digestion. Intrinsic factor is a necessary substance in order to actually increase the absorption of vitamin B12 in the small intestine. Chief cells are another structure inside the actual gastric pits, and these are going to secrete pepsinogen. Pepsinogen is converted into pepsin via the action of hydrochloric acid. Pepsin is important because it functions to digest and break down proteins into simpler amino acids. Enteroendocrine cells are actually going to be cells of the stomach that produce specialized hormones. In this case, G cells are an example of this, and they produce the hormone gastrin. Gastrin causes parietal and chief cells to release their products. So you can see it's this flowchart cascade process of hormonal cells responding to food coming into the stomach to thereby release gastrin. Gastrin thereby causes the release of hydrochloric acid and pepsinogen, and the combination of hydrochloric acid and pepsinogen together will make pepsin. So all of these are going to kind of interact with each other. Another type of cell that's not listed here is going to be the ECL cell. These produce different histamines and hormones that affect other cells in the digestive system. Now here we can see some other examples of the histology of the stomach, where we can actually see the parietal cells where they are. We can see the mucosal neck cells in the neck of the gastric pit, and we can see the G cells far deeper down next to the chief cells. Moving on from the stomach, we now enter into the intestinal phase of digestion. The intestinal phase is going to be where most of the digestion actually occurs, and this occurs in the small intestine. The large intestine, which follows the small intestine, is designed to concentrate waste for excretion. There is a lot of motility in the large intestine, and a mass movement moves the bolus forward to trigger the defecation reflex. So this is going to be moving and compacting the different food items that have been processed for later release. Digestion and absorption occurs in the large intestine, and diarrhea can cause dehydration. Now, the small intestine is going to be approximately 20 feet in length, and it is only 1 to 2 inches in diameter. It is longer than the large intestine, but smaller in diameter, hence why it is the small intestine. The small intestine consists of 3 parts. This is in order. It goes the duodenum, jejunum, and ileum. The duodenum is the smallest portion, and it is only about 10 inches long and receives digestive enzymes from the pancreas, bile, liver, and gallbladder. This is actually going to be an overall mixing bowl. It's going to serve as a mixing bowl for the different the chyme from the stomach and secretions from the pancreas and liver. The jejunum is 8 feet long. Now, most of the digestion is actually going to occur here. Most of digestion and absorption occurs in the jejunum. The first half of the jejunum has very prominent plicae and villi, but most diminish in size and number as you progress through the length of the jejunum as absorption decreases. Finally, we have the ileum. This is the longest segment. It's about 12 feet long, and it ends at the ileocecal sphincter, or valve. This would connect it directly to the large intestine. This in the ileum absorbs different bile salts, vitamin B12, and any remaining nutrients, but it doesn't have the same absorption rate as the jejunum. Here we can see a diagram and layout of the different structures, the blue being the duodenum, with the pink being the jejunum, and the peach or orange color being the ileum as it extends all its way down. Looking at a bit of the histology of the small intestine, we can note first the mucosa. Now, the mucosa forms a large number of finger-like projections called intestinal villi. These are going to project into the lumen. The apical portion of each intestinal villi exhibits numerous smaller microvilli. So the microvilli are going to extend off the villi. We can see the microvilli here. Now, the microvilli form what is called the brush border. The villi and microvilli are functioned to increase surface area. Now, this is very important in digestion for absorption. So it's allowing more spots for things to touch in order so that it can be absorbed. We also have different intestinal crypts here. Now these are similar to gastric pits. These are going to be deep pockets at the base of the villi that contain different stem cells to help constantly replace these columnar epithelial cells, because there is a lot of acid that's still going to be in this first part of the small intestine. Finally, we have the lamina propria, as can be seen here. Now, extending through the lamina propria of each villi is going to be a terminal lymphatic vessel called a lacteal. That's the central green vessel that we see around the capillary network. The lacteal is going to be very important in absorbing and transporting lipid protein complexes from the intestinal lumen through the lymph system. One of the major functions of the lymph system is to transport dietary fats, because they're a little bit too large initially for the actual blood vessels. This is going to then transport them to the liver for later processing. Here we can see another example of the brush border and all of those microvilli that extend off. The darker structure that you can see towards the left side of this slide is actually going to be the nucleus, and then it becomes slightly more clear. This is actually going to be some of the mucosal cells that we have throughout our intestinal tract that are designed to release mucus out onto the surface of this brush border. Now, moving down into the large intestine, we can see that it consists of several parts. All of them are really going to be related to being the colon. Now, the large intestine is 5 feet in length and 3 inches in diameter. It consists of the following regions. First is the cecum. The cecum is actually going to have the ileocecal valve and connect directly with the ileum. From there, we go up the ascending colon, across the transverse colon, down the descending colon. We bend slightly at the sigmoid and then exit via the rectum, which will connect to the anus and exit the body. Here we can see a diagram again of what I've talked about. The first structure, where the square is actually located on and zooming in on, is going to be the cecum. This is an expanded pouch that receives material from the ileum via the ileocecal valve or sphincter. The cecum stores material and begins the process of compaction. Also attached to the cecum externally is going to be your appendix, or the variform appendix. This is found on the posterior medial surface, and this hollow tube is approximately 3.5 inches long, but it can be variable. I've seen it longer. And it is largely a lymphatic tissue and serves an immune function similar to the tonsils, primarily protecting the large intestine from different bacterial infections. This, of course, can become infected, inflamed, and cause violent pains that will signal that it's ready to burst. And this inflammation is called appendicitis. And from here, you need to have your appendix removed through surgical means. As we extend up the colon is going to simply be the colon throughout, ascending, across the transverse, down the descending, and then to the sigmoid colon and rectum. Each of the bends is going to be a different flexure that we can see. Now, the ascending colon begins at the border with the cecum and ascends along the right posterior lateral abdominal cavity to the inferior surface of the liver, where it turns sharply to the left at the hepatic flexure, or the right coeliac flexure. This marks the beginning of the transverse colon. Movement of the material from the cecum to the transverse colon is very slow and involves peristalsis and haustral churning. Haustral churning is going to involve the different pouches that we see here called haustra. Every little part of this large intestine is composed of haustra. Haustra are these little pouches that are going to section off the bolus, which would be compacted later into fecal matter, and it's going to be bands of smooth muscle. That's why it forms these pouches and allows it to move food a little bit better. At the transverse colon, it's at the hepatic flexure, and the colon curves anteriorly and across the abdomen to the left of the anterior lateral side. It then makes a downward bend at the splenic or left coeliac flexure, and it becomes the descending colon. Movement of compacted material from the transverse colon through the rest of the large intestine is called mass movement. This occurs only a few times a day and is normally stimulated when the distension of the stomach and the duodenum happen. And this, of course, happens when you eat another meal. So mass movement will only occur when we eat another meal. The descending colon proceeds inferiorly along the left side of the abdomen until it reaches an S-shaped curve called the sigmoid colon. The sigmoid colon is a short S-shaped segment that begins at the sigmoid flexure and ends at the rectum. The rectum itself is going to be a short 6-inch expandable rectum that would thereby expand into the anal sphincters and then into the anus. So some histology of the large intestine. The walls are going to lack villi, but it has numerous goblet cells and very distinct intestinal crypts. It's designed to produce a lot of mucus and lubricate the overall passageways, as well as contain a large number of lymphoid nodules to protect against bacterial buildup. To review some of the order of the large intestine, we'll start back with the cecum, remembering that it is that large pouch and the site of where compaction begins. Attached to it is going to be the appendix, which functions as a lymphoid organ, and it's going to link itself to the ileum via the ileocecal valve. The colon is going to be the ascending, transverse, descending, and sigmoid. Here, remember that waste material goes up the ascending, around the hepatic flexure, across the transverse colon, around the splenic flexure, down the descending colon, around the sigmoid flexure, and then out through the sigmoid and into the rectum. The rectum is an expandable area about 6 inches long for temporary feces storage. Movement of the fecal material into the rectum triggers the urge to defecate. Now there are two major sphincters here. The anal sphincters are going to be a circular layer of muscle, and the inner anal sphincter layer is going to be smooth muscle that is going to form from the muscularis externa. The external anal sphincter is an outer layer of skeletal muscle and is voluntary. The anus is simply the exiting of the anal canal. Here the epidermis becomes keratinized. Here we can see the sigmoid colon as it leads down to the rectum, and we can see a diagram of the two sphincters as it enters into the anal canal. So the fecal matter would exit down, and we can see the muscularis externa is going to form the internal anal sphincter. On the outside of this is going to be the external anal sphincter, which is going to be a band of circular muscle fibers that stays contracted until is voluntarily relaxed during defecation. Now, the defecation reflex begins with the distension of the rectal wall after the arrival of feces. Here, it is going to have fecal matter that triggers the stretch receptors in the rectal wall, and this causes a relaxation of the internal anal sphincter where feces moves into the anal canal. Now, without the voluntary stimulus, the external anal sphincter stays contracted. Voluntary relaxation is required for the external anal sphincter to allow for defecation. So now we've gone through the primary digestive system. We're now going to look at some of the accessory digestive organs. Namely, we've talked about the salivary glands. We're now going to finish up with the gallbladder, pancreas, and liver. So we'll start with the liver. The liver is the largest visceral organ of the body, and the liver is involved in a number of functions, over 200, but there's a few important ones that we'll talk about. Metabolic regulation is going to be one of the main ones. It regulates circulating levels of glucose, amino acids, and fatty acids. It also absorbs vitamins. So it literally deals with all of the major macronutrients and vitamins that your body deals with. It is involved in hematological regulation, both the removal and destruction of red blood cells, as well as being a huge blood reservoir. It is the largest blood reservoir in the body, where blood moves through large hepatic sinusoids, and the liver also removes debris, pathogens, and damaged old red blood cells from the blood itself. The liver functions in the removal and destruction of toxins, and also in the synthesis and secretion of bile. Bile, which we'll get to a little bit more, is a watery substance containing ions, bilirubin, and bile salts. The ions in water help dilute and buffer the acidic chyme in the duodenum, and the bile salts are critical in helping digestive enzymes break down lipids into fatty acids. Here we can see the overall structure of the liver. The liver is divided into 4 lobes. The right and left lobes are very easily observed on the anterior surface. and they are separated by a part of the mesentery called the falciform ligament. This is not a true ligament. It is a remnant of the umbilical cord from a fetal development. A thickening of the inferior margin of the falciform ligament is the ligamentum teres. This is looked at as the round ligament, which is the remnants of the umbilical vein as it attaches in. The anterior surface is smooth, has a bare area, and follows the curve of the abdominal wall. The posterior surface has impressions of several visceral organs, namely the stomach, small and large intestine, and the right kidney. The caudate lobe, visible on the posterior surface, is bounded by the falciform ligament and the shallow impression housing the inferior vena cava that can be seen as that blue vessel on top. The small quadrate lobe lies inferiorly between the falciform ligament and the gallbladder. The hilum of the liver, similar to the hilum of the lungs, and later we'll see of the kidney, is the location of the hepatic portal vein, hepatic artery, and the common bile duct. So this creates the portal triad. Here we can see another view of the hilum of the liver, where the hepatic portal vein, hepatic artery, and the hepatic duct will actually exit the liver. Now the hepatic duct is going to be the green vessel that you see, and it extends down and connects to the cystic duct. The cystic duct is going to connect to the structure of the gallbladder, which we'll talk about in just a bit. The common hepatic duct becomes the common bile duct as it merges down with the cystic duct and then inserts itself into the pancreas. Now the cystic duct connects to the gallbladder. The gallbladder is a hollow, pear-shaped organ, and this is going to be involved in the storage and concentration of bile before excreting it into the small intestine. Bile will move from the gallbladder via the bile caniculi into the right and left hepatic ducts, to the common hepatic duct, and then into the gallbladder via the cystic duct, or the duodenum via the common bile duct. Bile in itself is a unique substance. Bile salts are going to break lipid droplets apart through a process called emulsification. Now, how it does this is based upon what it's composed of. Remember, the bile is a watery substance that contains ions, bilirubin, which is a breakdown product of hemoglobin from reabsorbed red blood cells, and bile salts, which are actually special lipids like detergents. So we actually see that a bile salt has a hydrophobic side and a hydrophilic side. So this, once mixed and emulsified, is going to help to separate a lot of these fat globules from each other, thereby allowing for the easier breakdown and absorption of them into the body. Here's going to be some of the internal structures that we can see of the gallbladder. The gallbladder would consist of the fundus, which is going to be the enlarged portion, the main body of it, which would be the primary storage site, and then the neck, which would lead out into the cystic duct. The cystic duct, remember, is going to combine with the common hepatic duct to form the common bile duct, which is going to insert directly into the pancreas. From the pancreas, you'll see them where they actually combine together. The common bile duct will merge with what is known as the pancreatic duct at a structure called the duodenal ampulla. This is a muscular chamber approximately halfway along the duodenum where the common bile duct and pancreatic duct empty their respective secretions. Almost all digestive enzymes are produced by the pancreas, which we'll get to in just a moment. The gallbladder releases the bile, and the pancreatic duct will release all of the other enzymes, and this is designed to neutralize the acidic chyme and to begin the digestive process. Now, the pancreas we've talked about back in the endocrine lectures is going to be involved in just 1% in the regulation of blood sugar, and that's going to be with the release of insulin and glucagon from the beta cells. The pancreas itself, though, remember, is primarily involved in exocrine function. The pancreas, as we've talked about, lies posterior to the stomach and extends laterally from the duodenum towards the spleen. It has the head, body, and tail, and a major structure in the center of it called the pancreatic duct. This is going to deliver the exocrine secretions that 99% of the pancreas is involved in. This overall secretion is called pancreatic juice. Pancreatic juice is a combination of water and ions secreted by the epithelial cells lining the duct, and exocrine secretions of different enzymes and buffers. It produces about 1,000 milliliters or 1 quart of this pancreatic juice each day. Here we can see a diagram of the pancreas and how it actually connects directly to the small intestine, where it's going to release its different compounds via the duodenal papillae.